To begin, let us acknowledge that Queen's University is situated on traditional Anishinaabe and Haudenosaunee territory. We are grateful to be able to live, learn, and play on these lands, and we thank the Indigenous peoples of this area for their care of their land for thousands of years, and we hope to honor and respect them as we hold our virtual event today. I'd also like to take this moment to personally thank everyone for attending our virtual presentation today. Our speaker this afternoon is Dr. Susan Cork, who'll be presenting on the ecology and epidemiology of Lyme disease in Western North America. Dr. Cork is a professor in ecosystem and public health at the University of Calgary in the Faculty of Veterinary Medicine. She joined the faculty in 2008 as the founding head of the Department in Ecosystem and Public Health. Prior to arriving to Canada, she spent 5 years with the New Zealand Ministry of Agriculture and Forestry, where she held a number of policy and management positions. She completed her veterinary degree at Massey University in New Zealand in 1987, and then worked for the Society for the Prevention of Cruelty to Animals in Fiji and in a private veterinary practice in the United Kingdom before returning to New Zealand to complete a PhD in the Department of Veterinary Pathology and Public Health. In 1995, she accepted the position of veterinary laboratory manager in Eastern Bhutan as part of the European Union funded project to strengthen veterinary services in Asia. In 2003, after 6 years in the United Kingdom working in government and academia, she returned to New Zealand to join the government service and obtained a diploma in public policy from the School of Government, Victoria University in Wellington. Since then, she has traveled widely in Asia and across the Americas and has returned to Bhutan a number of times. Her research interests are in global health, wildlife health, and vector-borne diseases, as well as climate change. We will have Dr. Cork present, and then we will open up for questions from the audience. You can ask your questions by entering in the chat box, or you can raise your hand using the icon, or if time allows, unmute if we still have time to ask questions. Please help me welcome Dr. Cork to the podium.
Thank you, Veronica. Can you hear me okay?
Perfect, and we can see your slides.
Excellent. So I'd like to share a series of slides with you that have been prepared by myself, my colleague Isabel Coulogne, and one of our summer students, Carl Dizon. This is very much an interdisciplinary multi-agency project, which we've been working on for 4 years. And I'll share the names of some of the excellent people we've been working with at the end of the presentation. But basically what I'm going to talk about is the ecology and epidemiology of Lyme disease and the tick vector, Ixodes pacificus, in Western North America. Okay, I think sorry about that. I'm going to have to get the other slideshow. Sorry about that. So here is our land acknowledgement from the University of Calgary, recently updated. I won't read it out, but obviously it's very important to acknowledge the various tribes that have been looking after this land for many, many years. And we really appreciate the fact that they've been here to do that and working with them on various projects currently. So a bit of a summary of the infection prevalence of Lyme disease across Canada. And most of you know that the prevalence is much higher in most parts of the eastern United States and Canada. But there are pockets of Lyme disease cases across the West as well. The epidemiology is very different because the primary tick vector is different, and we'll come on to that in a moment. But what we do want to point out is that the cases in the West, particularly in Western USA, are gradually increasing, not to the extent as they are in the East, but certainly things are changing, possibly a result of climate change in various populations of tick reservoirs. Now, one of the reasons, and you probably are familiar with this, why there is a difference is because the reservoir hosts are somewhat different for Ixodes scapularis in the East than what we see for Ixodes pacificus, which is the main competent vector in the West. And this little chap here, the mouse, is obviously very important in transmission dynamics for the Borrelia burgdorferi because it's immunotolerant. And so you get large levels of bacteria in the bloodstream. So when nymph and larval ticks bite this host, then they're more likely then to transmit the Borrelia to another host. And nymphs in particular are considered one of the higher likelihood of risk transmitting the Borrelia to human cases. Also, there's a lot of white-tailed deer in the eastern parts of Canada and North America. And whereas they are a bit of a dead-end host for the Borrelia itself, they can lead to large numbers of adult ticks developing in the environment. The white-tailed deer are not so common, of course, in the West. The black-tailed deer are thought to be more significant for tick dynamics in the West. If we look at past surveillance efforts, we can see here that Ixodes pacificus is pretty common in California, occurs in Oregon, Washington, and south coastal British Columbia. But we do believe that that range is expanding, and certainly early climate change projections in California show that it's expanding in that region. We can see also in the top table there that there's also Ixodes angustus and other Ixodes species which can transmit Borrelia in Western North America, but these are thought to be less important in transmission of Lyme to people because they're less likely to bite people. And so there's quite a lot of interactions between these different tick species, which does affect to some extent the likelihood of Borrelia being present and transmitted. And also these ticks can transmit other pathogens, including anaplasma and different types of Borrelia that aren't the same as burgdorferi. So the life cycle is not that different, I suppose, to what you see for scapularis. Typically has a 3-year life cycle in California, but it can be up to 4 years in Canada and the northern parts of the US. There's still quite a lot of research underway about this because it's not fully understood. But as with scapularis, each stage of the life cycle, the larva, the nymph, and the adult requires a blood meal. And you'll see a little picture of a reptile in that little life cycle there, and I'll come on to that in a minute. So this is part of a broader project which was funded by Public Health Agency of Canada, which is a project led by the British Columbia CDC, Erin Fraser, is the project lead, and the University of Calgary looked after the modelling component along with colleagues at the British Columbia CDC and also colleagues across Canada. So the general project was looking at Dermacentor ticks as well, particularly Dermacentor albopictus. And we also did some mapping and modeling for the Asian longhorn tick, should it arrive in Canada from the USA. But I'll talk today more about the Western black-legged tick, Ixodes pacificus. So Carl Dizon, who we had last year as a summer student, he developed a narrative literature review along with a number of our entomology colleagues and Isabel and myself. And then with Isabel, he developed some species distribution models to predict current and future distribution of the Ixodes pacificus tick. So fairly typical literature review. It wasn't a full scoping review. We targeted particular keywords, which are listed here, and then we divided it into several sections. One of the things that we put a lot of effort on is looking particularly at the hosts in different parts of Western North America. Now, what he found when he looked at the literature over a period of time is there's been a lot of research, some very good papers actually published from California. There was a lot of grey literature from Oregon and Washington, and there was some publications already out from British Columbia. But by far the majority of work that's peer-reviewed and published seems to be based in California. He developed some theories and hypotheses based on what he'd read, which we looked at when we started doing the modelling work. If you want to read the review, it was published fairly recently and it's available open access online. What I'd like to point out actually with that review, we had 3 very thorough reviewers, I think 2 of them likely from the US, who were very, very helpful. And they were really keen to see the paper come out because they felt there hadn't been a lot of attention on what's going on in Western North America, largely because the levels of Lyme disease are smaller. And so it's been harder to get funding for that. But nevertheless, some really interesting studies on the ecological side. So looking at the maps that have been previously published, we can see the one at the top there is just showing Vancouver Island and parts of British Columbia, the darker areas are where you're more likely to find the Ixodes pacificus ticks. And then lower down, you can see some of the work that's been published from California. If you want to know more about the clinical aspects of Lyme disease in British Columbia in particular, I recommend you go to the British Columbia CDC website. They've got some really good tools and information, plus a number of maps available there, which will probably be updating fairly soon based on the work of that project team. So if we look at the key hosts in California, what kept coming up was this western fence lizard. The larval and nymph stages really prefer to feed on this, although they do feed on squirrels and chipmunks and mice as well. But what's been found quite recently, and some of it's been published, some of it's still in the pipeline, is that when ticks feed on this western fence lizard, it seems that they have an anti-Borrelia component, and it's not fully understood what's going on. There is some lab research going on to find this out right now, but it seems that if the ticks bite this lizard, even if they have Borrelia, that the Borrelia doesn't do very well. And so if ticks then bite a lizard that's already been bitten, that they don't actually get the Borrelia, and so they don't transmit it to other species. So this is quite an interesting finding, but it shows that if you have a lot of these western fence lizards in your biodiversity in a particular region, then other hosts play less of an important role, even though it is found that squirrels and possibly the chipmunks are almost acting as reservoir hosts, but with sort of a smaller number in many of the coastal regions where the Borrelia is quite commonly found. Then when the ticks go into that community, then there's less likelihood of them biting these rodents. They prefer to bite the lizards. Now, that's not the same across California. It varies from north to south, and there's further work ongoing. And then, as I mentioned, the white-tailed deer isn't such a big species in that region. It's more the black-tailed deer. If you go further north, and particularly into British Columbia, then this northern alligator lizard is important in the life cycle. And we found a very interesting old thesis, and I don't believe a lot of the work's been published. It was a master's thesis, Arneson, and he had done some quite interesting field work and found a lot of larval and nymphal stages of Ixodes pacificus feeding on this particular host. There hasn't been a lot of laboratory studies done on this particular host, but it's possible that it has the same bactericidal effect as what happens when the western fence lizard is present in the environment. If you look at the habitat and the range of this particular lizard, it's pretty widespread. So at the moment, we haven't actually included this host data in our models, but it might be something that we would do in the future. We just need to do a few more collections in some of the areas where the Lyme disease is less likely to be found. A lot of our models, we used Maxent, which is a machine learning algorithm. And the reason why we picked that, even though we tried lots of other methods, is because it's quite reliable with presence-only data, which is basically what we were dealing with for the tick locations. If we look at the preferred environmental and climatic factors, as with Ixodes scapularis, it does like woodland areas, but what is quite interesting, even though it likes to be protected in leaf litter, is it's also found quite often on the coastal regions, which was surprising, actually, when they first found it there. But certainly, even in Arneson's work, he found that that was common too in British Columbia. So to develop our models, we needed, obviously, tick locations and confirmed identity of ticks. We also looked at identified Borrelia in the ticks that were submitted, but there's a lot fewer points. I will show you some maps for that, but that's still work in progress. We used Climate North America for our seasonal bioclimatic variables and EPA for our ecoregions. And then when we actually looked at the future climate, we sort of picked two. We did the middle of the road, the SSP 245 and the SSP 585. We didn't go to the very severe extremes, and we didn't project too far out beyond 2050, largely because our expert climate advisors said that that was so unpredictable. There's a lot of uncertainty in these modeling variables, of course. So the SSP is the shared socioeconomic pathway, and it takes into account various things that are going on in social and demographic activities and behaviours as far as what might happen with regard to reducing greenhouse gas emissions. So ecoregions, for anybody that isn't familiar with that, basically it takes a lot of things into account. You can use it to some extent to predict the flora and fauna of a region, but it isn't specific enough to know exactly what will be happening with particular hosts or very specific environmental factors. When you try and put the climatic variables for the future, there's a lot of uncertainty as to how the ecoregions might change, and a lot of modelling studies run into that dilemma. At the end of the day, models are only as good as the information you put into them, but when you actually look at the mathematical component and then you overlay the most likely selection based on what you know from a biological point of view, then you can end up with a pretty sensible prediction. And that's why these studies have to be very multidisciplinary. So the data that we were able to access came from the British Columbia CDC and also Washington Health Department. Our colleague Liz Dijkstra was very much engaged in this work, and we decided to start with just using active surveillance data, and that's because it gave us actual points where the ticks were found, and we didn't have this big question mark over travel history, which isn't always present when you look at passive data. But we also looked at the passive data later on, and I'll show you that. So using Maxent, I won't go into the details. One of my colleagues, Isabel Calonia, is in the audience. So if anybody has any specific questions about the modelling, she'll be very happy to answer those as well. So we used a training set and a testing set of the data. We used presence points, and then we generated pseudo-absence points as well. I won't go into this in any great depth, but there's a number of different stages to pick your best model. And initially, of course, this is based on mathematical fit. When we actually developed our maps, though, and our various models, these were the two that were the most suitable. And largely, we're looking at frost-free days, we're looking at moisture indices, ecoregion, and minimum temperatures at particular times of the year. This is all going to be published, and so it'll be available later on. But again, these slides will be available, and Isabel will be quite happy to answer specific details of the methodology if you're interested. But these two models basically are showing us something very similar. And the darker areas, of course, are where we predict the most likelihood of finding the ticks. Now, I might point out at this moment in time, however, it doesn't necessarily mean to say those ticks are carrying Borrelia. It means it's possible there's ticks there, they may have Borrelia, they may be considered risk areas, but that's extrapolating somewhat. And although you have to do that sometimes, we'd have to sort of note that the number of, or the relative percentage of Ixodes pacificus that are positive Borrelia is still relatively small compared to what we find for Ixodes pacificus in the East. Looking at our future projections, there's a number of different ways of doing this, but we talked a lot with our climatology experts, and these are the scenarios that we selected, and they seem very much in line with other published work. So if we look at the future, if we're looking into sort of mid-2050, then these are the sort of projections we might have. So we can see in the middle of the road scenario versus the very little control of greenhouse gases, there's going to be a change either way, but certainly there's a higher change, a higher percentage of change of habitat extension for the tick in the more extreme scenario. And there's a number of factors that might impact that. But again, we have to remind ourselves that we don't know entirely what's going to happen with the ecoregions. For instance, if there's a lot of fires, that's going to impact the ecoregions quite significantly, as would be the case if there's a lot of flooding. Now, these are the maps that we've developed recently for our knowledge translation component of our project with the BCCDC. We're still fine-tuning these, but essentially just to remind us of the predictors of interest, number of frost-free days is important, moisture indices, moisture deficit, there's a little bit of variation depending on which of those you pick because they're calculated differently. And then obviously the ecoregion is relevant and the mean warmest month. If we project that into the future, with our middle of the range, you can see some expansion, particularly in the valleys. And we do see, if you look at some of the ETIC submissions, even though we don't always know the travel history, it is available, and we might look into that. But there are submissions from places that are in the valley areas, and also some coming in from further up north, where we can predict there may be ticks in the future. But certainly there's some quite interesting activity going on. So if we're looking at mapping Lyme disease risk, and as I say, that's just indicative, because even if we find Borrelia in the ticks, it doesn't necessarily mean they're transmitting it, but they're kind of hotspots here. And this is based on the active sampling submission, so actually ticks that were sampled. And then if we look at the passive sampling data, we can see the active sampling data at the bottom and our habitat suitability models. But then if you see some of the passive data, which has been collected over many years by the British Columbia CDC, there's some slight differences in where we're finding tick submissions. So again, that's something that we'd like to look into moving forward. And our colleagues at the BC CDC are already working on that. And then mapping Lyme disease hotspots. Again, this is passive data, samples submitted from a number of different species, particularly companion animals. And again, these are in areas where we might expect to find them, so no great surprises there. And again, more information is available on the BC CDC website. Anybody that wants to be involved, obviously ETIC, most of you will be familiar with. There was a very good talk earlier in this week about that. And then iNaturalist we used a lot, because obviously ticks don't respect borders. And so we want to work further with our colleagues in the US, which a lot of you will be doing anyway, because looking at their findings and where they're predicting things is quite an interesting marker point to where we might like to extend surveillance and collaborations in the future. Now, the other thing, and again, this was mentioned earlier this week when we were talking about ETIC data, is there's something very interesting happening in Alberta, not with Ixodes pacificus, but with Ixodes scapularis. Now, the theory has always been that birds were dropped off in Alberta with migratory birds coming across from the east, and that these populations of ticks were really just sort of accidental. The ticks weren't established, and that's likely to still be the case. However, there are some interesting findings over the last few years, and there seems to be a few hotspots up around Edmonton. So we are working with the municipality up there, looking further into that, and we've talked to our colleagues at Alberta Health Services as well. They're interested in this because things are changing. It is a good time to actually start doing a little bit more examination of what's actually happening. And so we do actually have, obviously, keeping an eye on data that's coming in passively, but we also have some active collection, working with the Canadian Lyme Disease Research Group. We do some sampling in Red Deer and Lethbridge, our colleague Sean Durgos, who's on the call now. And we've also done some work in Calgary, Cochrane and Edmonton. And we're doing that every year in the tick season. We haven't really found a lot yet, but there are some things that we're investigating further. And we do have a summer student, Brittany Hammerlindell, who's working with us this year. And we're going to be working with ornithologists to look carefully at some of the bird migration data, looking at tick findings, and just seeing what might be going on. There are some very active banding projects across the province. Early stages yet, but we are hoping to look into this a little bit further. So acknowledgements, a huge team. Sylvia Checkley, epidemiologist here at the University of Calgary. Sean, who I think's on this call, entomologist at Lethbridge. Mohammed Morshed and his team at the BC CDC. Sunny Mack, who I think is also on the call. Elizabeth Dijkstra from Washington Health Department. Trevor Murdoch and Colin Mahoney, who gave us a lot of really excellent advice on the climate modelling side. Again, it's often having to make decisions each stage about which variables you're going to use, but certainly what we've found in our mapping very much aligns with what other researchers have found in their projects, particularly those based in California. So a lot more work to be done, but a very interesting project and a great team to work with. So thank you so much for your time, everybody.
Thank you so much, Dr. Cork, for your presentation. We're going to move on to the question and answers segment. So if you have any questions, please enter them in the chat box, or you can raise your hand icon and we will start the questions. I'm just checking the chat box right now. I don't see any questions in the chat box. Does anyone have a question from the audience? Go ahead, Dr. Vordu.
Yeah, hi. Can you hear me, Susan?
I can, yes, thank you.
Okay, great talk, very interesting. I wanted to ask you about the Ixodes scapularis that ETIC is picking up in Edmonton, and I was interested to see, I mean, we don't nearly get as many in Saskatchewan, but we also have this sort of pattern that you get some sort of in May, June, then nothing, not much in July, August, or nothing, and then the fall. And first, I just want to establish, are most of those ticks, I guess they're all probably adult ticks, right? Adult Ixodes scapularis?
The majority are, but Jade, I see, is on the call. She talked a lot about this earlier in the week. Do you want to comment about that?
Hi, Martin. That's a good question. It's mostly adults. I do believe we had a few nymphs last year, and as we're just at the beginning of the season, so this year the spring has been much busier than usual. So I would need to go back to see what they are. So you're still talking about what's been found in Alberta, correct?
Yeah, in Alberta.
Yeah, because the contrast for me with Saskatchewan is quite, it's really pronounced. So definitely there's something different going on. But one thing to remember, the other difference with Alberta is that unlike the eastern provinces, almost all of the records are from animals, whereby out east, it's at least 50% from humans. So there is something different happening with those. This set of records is different than what we get out east, but there's definitely more abundance than what we see in Saskatchewan. So mostly adults, but not only adults.
Well, what I was going to say is, you know, I can understand the fall peak because, well, first of all, we should say that, you know, adult ticks generally don't like to feed on birds, right? So it's larvae and nymphs that attach to birds, and so we would expect the nymphs to be getting dropped off in the spring, and then they would molt into adult ticks, and then that would make sense that you would see those adult ticks in the fall. But it's interesting that the adult ticks that you see questing in those earlier months, like April, May, June, that suggests to me they would have been dropped off the previous year as engorged nymphs, molted, and then overwintered and started questing then, you know, that early spring. So I'm just wondering about that, Susan, and also Jade, if you guys think that, you know, the early peak of adult ticks questing in Alberta, does that suggest to you that they're, you know, being dropped off as engorged nymphs the previous year and overwintering and then questing again that spring?
I think it suggests that this is what could be happening. But for me, what it mostly suggests is that there needs to be a bit more active work. So the kind of work that Susan was talking about. So the ETIC data is a bit more anecdotal. It doesn't give you a great, well, it's not anecdotal, but it doesn't give you a great snapshot of what's going on in the whole area. So I think active surveillance would probably allow us to figure that out. But I do find these bunch of adult records in the spring to be odd, and this year more than the previous years.
Yeah, because even if they were dropped off as adults, which is unlikely, then, you know, they just got a blood meal. So why would they be questing again, right? I mean, they should be. So yeah, it's very interesting. Thanks.
Thank you, Dr. Bordeaux.
Thanks, Marty. Yeah, we do find it very interesting. Obviously, there's been a particular understanding over many years. So if things are changing, we want to understand exactly what is changing before we jump to any conclusions. But there seems to be two peaks, and Isabel created the slide there from ETIC, but there is a peak earlier in the tick season, and then there's another peak in the kind of autumn time. So it does indicate something's going on. I mean, Sean, you're here on the call being our entomology expert. I don't know if you've got any thoughts or comments about that. We'll ask him later, but he will be involved in our summer project.
Sorry, I was just having trouble. I'm actually out in the field.
Oh, there you
We're
are.
doing spider and sampling right now. So, but yeah, no, I don't know. I don't have anything really to add based on the observations everybody else has made. So it's also interesting that Edmonton is kind of a hot spot there as far as reports. Their habitat is quite a bit different than Southern Alberta, which is a lot drier and stuff. And with Edmonton, whether or not that's a more suitable area for, birds and then into the forested areas. Of course, it's a little cooler and stuff too, but I don't know about the interaction between the temperature, but then also the much more humid and forested areas too. So lots of work to be done.
All right, thanks for all your answers. Appreciate it.
Thank you. We have a question in the chat. It's a two-part question. What is the infection rate of the Western tick? And are the co-infections they carry similar to Ecology scapularis?
It varies actually across different parts of Western North America. It kind of ranges between about 0.9 to 4.2%, but more often being about, the lower end. But there hasn't, because it's harder to get funding for something that's less of a problem. Not as many ticks are tested as people would like to see tested. Now, Washington Public Health, they did quite a lot of testing a few years ago, and we're still waiting for the most recent Borrelia testing results of ticks. But what we are told, I don't know that this has been fully published as yet, but the percentage of ticks testing positive is increasing. Not exponentially, but they're finding more that are positive than they did in the past.
Thank you. Thank you for the question.
The other part, actually, sorry, I didn't answer it.
Go ahead.
The other pathogens that it can transmit, yes, they are fairly similar, but again at lower rates. But we seem to have a wider diversity of tick species, Ixoides species, and different types of Ixoides have higher or lower likelihoods of having things like anaplasma, for instance. But again, there needs to be more work to know for sure. There's a bit published, but not a lot.
Thank you. Do we have any more questions from the audience? I don't see any other questions in the chat. If you have a question, now's your chance to ask Dr. Cork before we have to sign off in a little bit. Oh, go ahead, Dr. Bordeaux.
Yeah, I have one more question, and just based on what Sean was saying with Edmonton having maybe more deciduous trees and being a bit of a better habitat for Ixodes scapularis. So I know maybe you haven't done the climate modeling, but I'm just curious, would Edmonton be a good habitat? Is it much colder there compared to like Eastern Canada where Ixodes scapularis is doing very well, or are there sites in Eastern Canada where there's lots of black-legged ticks established that would be very similar to Edmonton? I'm curious whether you have any thoughts about that.
Well, Patrick Leighton and his team have been doing some models, largely mechanistic models. So there has been some work. Nick Ogden, I think, and Tim Lissick looked a little bit at these kinds of things. What's interesting, though, we have a colleague at the University of Calgary that's working on mosquitoes. And certainly the populations of mosquitoes of interest are much higher, much bigger around Edmonton as well. And what our colleague was saying is that his thought was it's more likely to be because it's moister up there. Calgary and south of that are much drier. Isabel, you've been looking into this. Do you want to add anything?
Yeah, it's the same thing that you were saying, that coming from, well, what we've seen also with the mosquitoes and everything is that, yes, it is slightly more humid and a little bit less warm, but it's changing. And here also in Calgary, we've seen in recent years that it's getting also humider, if it's the right word. So we'll see, but yeah, we didn't do any model yet. That's the plan for this summer. So we'll see what's going to happen. But yes, it is definitely less warm and more humid in Edmonton. And that's why also we're seeing more mosquitoes there than in here. So it's probably why.
Okay. Yeah, thanks. Yeah, that's sort of always, well, that's what I tell people, and I never know quite if I'm right, but I say, there's not we don't have very many black-legged ticks in Saskatchewan. They're not established here. And yes, it's cold, but they also have that very important moisture requirement, right? They're quite sensitive to drying out. And I always imagine that that's probably a big reason why we don't have established populations in Saskatchewan. But then, yeah, if you say that Edmonton is a lot more humid than southern Alberta, then maybe that could explain perhaps why populations might eventually establish there or are establishing themselves as we speak. Yeah, thanks.
Thank you. We do have a question in the chat. Thanks for an interesting presentation. I don't know much about BC's geography climate, but I'm curious if there's something particularly different about the north of Graham Island. It is a very distinct area coming up in the models.
It is quite different up there. They have found quite a lot of Ixodes angustus up there. Sonny Mack is on the call here. I don't know if he's able to say anything because he did the earlier modeling around this area.
Hi, yeah, so Graham Island is that the Hattagwai area. There was sampling done there by Dr. Morshed and his team. We did not find Pacificus at that time, but angustus was detected. In terms of climate, it's of the coastal regions of British Columbia, they're largely moderated. Temperatures are moderated by the ocean effect. And so even though it's further north in terms of latitude, the climate is relatively moderate and similar to even the southern parts of the BC coast. And so I guess we have a couple of things happening. One is the effect of migration, right? So as ticks are transported from the south to the north through adventitious ticks or even through history, you have these physical barriers. And so that could potentially be a reason why those tick populations in the north have not been quite as well established yet.
Thank you. Just checking to see, are there any additional questions before we wrap today's presentation up? I don't see any in the chat, nor any hand icons up. Last opportunity to ask Dr. Cork. Oh, give me 1 second. I see something in the chat. I think there's a reference to a paper regarding to ticks, hardy survivors that someone has put in. I don't see a particular question. If there is a question, please raise your hand.
I think Rob's making a great point that, ticks could evolve, right? We're always making these ecological niche models based on what they're currently capable of, but it's not impossible to imagine that Ixodes scapularis might evolve more cold hardiness and more resistance to dry conditions and be able to invade habitats that we currently think they're not capable of invading. So that's a great point.
Yeah, that is correct, because we've seen that especially with the Asian Northern tick, that if we compare the one with, or in Asia, but the one we know more in New Zealand, that tick is not acting the same way in North America that in New Zealand, for example. So they do adapt, and that is also a very good point.
Thank you. Rob, do you want to follow up with anything? I see you've turned your camera on for us.
No, that sounded fine. That's where I was going with this. The models may not jibe with the actual biology of the tick. And they've already shown that in Northern Ontario, where there's extreme temperature ranges, I can picture Nova Scotia here in the South Shore being ideal habitat, high moisture. And with climate change, we don't get the winters we used to, but to find them in Kenora, Ontario, at minus 40 degrees to plus 40 degrees, that's a huge range. So some of the models might be a little too limiting. These bugs can extend beyond where people have predicted, which is not good news.
Thanks, Rob. Thanks, Isabel and Dr. Bordeaux. Okay, Looking at the time, any final question for Dr. Cork before we wrap up today's presentation? Okay, seeing none. Well, I want to personally thank Dr. Cork for her presentation this afternoon. I also want to thank everybody for attending today's event. I'm just going to share with you right now just a slide that just talks about our presentations. We are continuing next week. We have 4 presentations lined up Monday through Thursday. The first is on Monday. We have Hannah Caird, Dr. Aaron Fraser, Stefan Iwezoa, and Quinn Stewart, who will be presenting on trends in tick surveillance and tick-borne diseases in British Columbia. That presentation is at 1 o'clock Eastern Time. Also a reminder to take part in our challenge, wear green, take a photo and share it with us to help spread awareness of Lyme disease and tick-borne diseases. You can also send in photos that capture your creative artwork, whether it's poetry, whether it's pottery, whether it's paintings or drawings. All photos submitted will be automatically entered into our draw for a chance of one of 4 $25 Starbucks e-gift cards. Our live draw will take place during our last presentation at 1 o'clock on May 31st, and you can send your photos into clyden@gmail.com along with your name and email. And finally, just a pitch that the TickNet Canada will be having its first in-person scientific symposium this fall in Toronto, Ontario, October 24th and 25th. We're hoping to get registration and abstract submission open soon. We're aiming for June 1st. The planning committee has been working diligently over the last several weeks, and we hope to have more news as the week goes on throughout May, and hopefully be able to share towards the end of this month a registration link. And of course, if you do want to receive updates from our network related to events or newsletters, and especially around symposiums or these Lyme events, you can always get added to our distribution list. Just send an email to clyden@gmail.com and we'll be happy to add you to our distribution list. Again, thank you to Dr. Cork for today's presentation. Thank you for everybody for attending, and we hope to see everybody next week as we continue our talks. Take care and have a great weekend, everybody.
Thank you. Bye.